Hi everybody, in this video we are going to discuss about pollen pistil interaction. Pollen pistil interaction from chapter reproduction in flowering plants. Reproduction in flowering plants. Here pollen pistil interaction, before we talk about this, we also should know first the simple word, the pollination, which we already discussed. What is pollination? It is the transfer of pollen grains. It is the transfer of pollen grains from anther to the stigma. From anther to the stigma of a flower. But here we have to remember that pollination, the pollen grains that are falling on the stigma, okay, all the pollen grains that are falling on the stigma may not be the right type. Which means there may be many other different types of pollen grains. Maybe other types of pollen grains which also fall on the stigma. It may not be belong to the same species. It may not be the pollen grains that are falling on the stigma may not be of the same species. So pollination does not guarantee the transfer of right type of pollens. Pollination does not guarantee the right type of pollination transfer. So there is no guarantee that compulsorily the same type of pollen grains will be falling on the stigma of the same species. So based on that, we can come across that there are three different types of pollen grains may be falling on the stigma. One, we can say compatible. One, we can say it as compatible. Here, compatible means right type. Compatible means right type. That which will be useful or which will be involved. That these pollen grains will be going for fertilization, will be involved in the process of fertilization. So, compatible, the first type of pollen grains. The second type of pollen grains, we can say them as incompatible. Incompatible means what? Wrong type of pollen grains. Wrong type of pollen grains. And the third type of pollen grains can be self-incompatible. Self-incompatible. This is also what? It is also wrong type only. It is also wrong type only, but this type self-incompatible is based upon the genetically. Genetically we are going to discuss that. So here we should remember there are chances that different types of pollen grains will be falling on the stigma. So that's why what we are saying, all the types of pollen grains that are transferred onto the stigma do not guarantee pollination. What is that? So first one we are saying it as compatible. Compatible means what? Right type we said. Right type means it is these pollen grains which will be involved in the process of fertilization, forming a pollen tube. So let us say that this one, the first one, as the compatible one. And the next one is incompatible, wrong type. Wrong type means what? The pollen grains of other species. It is not of belonging to this species. So because there are various agents, either it is wind or water, abiotic things or biotic things involving many types of animals, they may be carrying different visiting. These abiotic agents or biotic agents, especially biotic agents, when they visit different types of flowers, their body will be sticked with the pollen grains and they visit again different types of species. So when they visit, they may bring up wrong type, that is what? Incompatible pollen grains falling on the stigma. And this wrong type means what? Of, let us say, these are the second one which are of other species. Let us say that these are totally of other species. There is no choice of these pollen grains to germinate, pollen germination and pollen tube to form at all. And the next one is, the third type is called as the self-incompatible. This is also called as self-sterility. Which means what? Wrong type. It is based upon genes. Okay, it totally depends upon the genes. Which means self-incompatible. Self, the word self here indicates that these pollen grains are of the same flower. These pollen grains that are these pollen grains are of the same flower. But what this flower is doing, it is not promoting the self-pollination. It will not allow these pollen grains to germinate and form the pollen tube. So that is called as what? Self-incompatible. It is totally under the control of genes that are present in that flower or in that plant, which will not allow its own pollen grains to germinate and fertilize the egg that is there present in the ovary or in the ovule we say, or otherwise embryo sac consisting the egg. So here we have to remember that pollination do not guarantee the transfer of right type of pollen grains. Pollination do not guarantee the right type of transfer of pollen grains. Okay, so here, which means that there may be any type of pollen grain that will be falling on the stigma, but here which one will be germinating? Which one will be germinating and entering into the ovule is the question now. Okay, so which one we said it is the compatible one. We already said it is the right type, we are calling it as. So right type of pollen grains will enter into the ovule, to fertilize the egg. So here it is now the pistil, when it is interacting with the pollen grains that are falling. Okay, the continuous dialogue, the continuous interaction between the pistil and the pollen grain makes the identification of whether it is compatible, incompatible or self-incompatible. So that's what we are calling it as pollen-pistil interaction, or it is the pistil which is having the ability. Pistil, this is the pistil now. Pistil is having the ability to identify the compatible ones, means right type of pollen grains. So it makes the pollen grain to germinate and produce the pollen tube, carry the male gametes to enter into the ovule and fertilize the egg that is present in the embryo sac. So we have to remember that it is the pistil which is having the ability to identify whether that pollen grain that is falling on the stigma is right type or wrong type, or it is wrong type when we are saying including self-incompatible. So here we should remember, the first point is that we have to remember is that pistil is having the ability to identify the compatible pollen grains. Once it identifies, this process leads to post-fertilization events. So once it identified, the pistil identifies the pollen grains. It is interacting with the pollen grains continuously. So once it identifies, it leads to what? Post-fertilization events. Okay, post-pollination events. What are those post-pollination events? That is leading to fertilization. So here, what we have to remember is that when the pistil is interacting continuously with the pollen grains, it leads to post-pollination events. That leads to fertilization. And what if they are wrong type? We are saying that they are incompatible or self-incompatible. If it is incompatible or self-incompatible, what will happen to these two types? They will be stopped. They will be rejected. They are not accepted. They are rejected, which means there will be no formation in these two. There will be no formation of the pollen germination. There is no formation of the pollen germination and also pollen tube. Okay, pollen tube will also not form in the incompatible and self-incompatible conditions. When the pollen grains are rejected, they will not grow. Here if you are seeing, it is completely from other species, so it will not germinate at all. And if it is self-incompatible, even if it is germinating, even though it is producing the pollen tube, at certain period of time, it may be completely, totally destroyed. So this continuous dialogue between the pollen grains and the pistil is under the control of various types of components, means chemicals. There may be different types of chemicals that will be always interacting between these two. Which one is the right type, which one is the wrong type, it will be under the control of various types of components, which means different types of chemicals that are being identified by the botanists who are discovering and doing research on this to identify what are those different types of chemicals which are helping in pollen pistil interaction. Pollen pistil interaction. So we said that here the right type of pollen grains will be germinating and producing the pollen tube. The right type of pollen grains will be germinating on the stigma and produces the pollen tube carrying the male gametes. So how this process will be taking place? How this process is taking place? As we all know that when we take a pollen grain, okay, if you are taking the pollen grain, once this pollen grain falls on the stigma, when the pollen grain falls on the stigma, here we should know few points regarding the pollen grain. We already know that pollen grain is of two layers. It is of two layers, we say. So suppose it is having the outer wall and also the inner wall. So what is the outer wall and inner wall called as? The outer wall is called as the exine, we are calling it as, and the inner wall is called as intine. Exine and intine. These two are the walls of the pollen grain. Walls of the pollen grain. Inside the pollen grain, there will be two cells. There will be two cells. One is very big cell. Okay. This is the vegetative cell, we call it as vegetative cell, which is rich in food material, reserved food material, and having irregular nucleus. It has a irregular nucleus that will be present. And please remember, this vegetative nucleus is rich in what? The reserved food material. Reserved food material having a irregular nucleus. And inside this vegetative nucleus, we will see one spindle-shaped cell that is present. We will see one spindle-shaped cell that is present inside the vegetative cell. This cell is nothing but the generative cell, we are calling it as. This cell is called as what? The generative cell. It is called as the generative cell. Having the nucleus. Now it is in spindle shape. So this cell is in what shape? This cell is in spindle shape. This is in spindle shaped in form. Okay, in shape. Now this is the pollen grain now. So this pollen grain, wherever there is no exine. See, if you are seeing here, say as we said that this is the pollen grain, here is the exine, and here and there is no exine. There is no exine. There is a small thin wall exposed of what? The intine wall. Means the exine is not continuous. The exine is not continuous. So this area where there is no exine is called as what? The germ pore, we are calling it as. We are calling it as the germ pore. Otherwise we can say here only it is called as what? The germ pore. So what is this germ pore? It is the place. Germ pore is the place where there is no what? Exine. It is only having the thin wall called as what? The intine. So when these pollen grains, we are talking about the compatible pollen grains, right type of pollen grains. Okay, so when this pollen grain falls here, the right type of pollen grain which will be producing the pollen tube when it undergoes germination. So the right type of pollen grain, as soon as it falls on the stigma, so suppose if this is the stigma, let us say that this is the stigma now. So once this pollen grain falls here, there are some points that we have to keep in mind here. As soon as this pollen grain falls on the stigma, this stigma will be producing or will also contains what? Water. It also contains sugars. Okay, it also contains sugars or organic acids. Okay, organic acids we say. Organic acids. So because of this, now what happens is that this pollen grain will absorb. Okay, the pollen grains that are falling on the stigma now absorbs this water and even the sugars or the organic acids and starts enlarge in size. They will be bulged in size. So we already said that pollen grain is of two walls, exine and intine. So when it is expanding, the inner wall that is called as the intine, where it is exposed through an opening called as what? The opening is called as what? Germ pore. Here, this this pollen grain starts producing a pollen tube. It starts producing a pollen tube. So from where the pollen tube is formed? Germ pore. What is this germ pore? It is the place where there is only the intine. Okay, it is a place where there is only intine. So please remember, 2 points are very important. We are seeing here there are many germ pores. Here this is also called as the germ pore. Here this area is also called as the germ pore, which means the pollen grains may consist of many germ pores. Many germ pores. But most of the times, it is through one germ pore only the pollen tube will be formed. It is only through one germ pore the pollen tube will be formed. If it is formed from only one germ pore, it is called as monosiphonous. It is called as monosiphonous. Okay, it is called as monosiphonous. If this is seen very clearly in Poaceae, it is seen in very clearly Poaceae family. If it is, sometimes the pollen grain may produce many pollen tubes. Whenever it is producing many pollen tubes, we call it as polysiphonous. We call it as polysiphonous. So what is polysiphonous? When the pollen grain is producing many pollen tubes, we are calling it as polysiphonous. What is monosiphonous? When the pollen grain is producing only one pollen tube, it is called as monosiphonous. So here polysiphonous. Here this is the best examples are, it can be seen in Malvaceae family. It can be seen in Malvaceae family and Cucurbitaceae. Okay, in Cucurbitaceae we can also see this formation of polysiphonous, means producing many pollen tubes from single pollen grain. But most of the times it is one pollen tube that is produced carrying the male gametes. So here what we have learned, now as soon as the right type of pollen grain falls on the stigma, as soon as the right type of pollen grains falls on the stigma, it starts germinating and produces a pollen tube. If it is wrong type, if it is of other species, totally it will be rejected. If it is of the same type, okay, self-incompatible we are saying, it may germinate, but it cannot produce the pollen tube continuously, and because it is under the control of genes, and it will be destroyed. That we said as self-incompatible. So here what we have learned, polysiphonous, monosiphonous and polysiphonous. So now slowly the pollen tube starts germinating and producing the pollen tube. As this pollen tube germinates and produces the pollen tube, so here we can say that this is the pollen tube. Okay, this is the pollen tube. So when the pollen tube is germinating, we already said what? That it has two cells, vegetative cell and generative cell. So these two cells, first, this wall is of what? The intine. First, the vegetative cell will be coming off. Then followed by what? The generative cell. And this is vegetative cell, and followed by what? The generative cell starts coming. Sometimes this particular style, okay, we said that this is stigma, the flat landing part of the pistil is the stigma, and the cylindrical part we already studied, which we are calling it as what? Style. This style may be hollow, okay, it may be hollow or solid. So when we say that this is hollow or solid, and please remember, the pollen tube has to penetrate through the cells. Okay, when we say that it is, especially when it is solid. Now, whenever we say that the style is solid, it is consisting of parenchymatous cells. Okay, it is consisting of this region. Okay, this region consists of parenchyma cells. Okay, parenchyma cells. So these cells are packed together, and through which now what should germinate? What should now move on? It is the pollen tube. It has to move on, and please remember, this pollen tube will release some enzymes. It will release some enzymes to digest the cells of the style. To digest means to break the cells, to make a way and move forward towards the ovule. So what this pollen tube is doing, from the tip of the pollen tube, the tip of the pollen tube releases certain enzymes which will digest the cells of the style. Okay, which will digest the cells of the style. If the style is what? Solid. So now what is happening? The pollen tube is moving, the tip of the pollen tube is releasing the enzymes, breaking those cells, and it starts making a way. At the same time, when it is breaking up the cells of the style, it also absorbs the food material from it for its growth and development. It will be absorbing the food from these cells of the style and starts moving on. And as this elongates, it starts moving and moves into the ovule. Okay, it enters into the ovule that is present in the ovary. That is present in the ovary. So here we have said that the pollen tube is carrying the vegetative cell and also the generative cell. Vegetative cell and generative cell. And as this pollen tube is extending, now we have seen that all that which is there in this pollen grain have moved into the pollen tube. What is moved? The vegetative cell along with its cytoplasm and the generative cell will also be there present in the tip of the pollen tube. Tip of the pollen tube. As it is moving, we should keep in mind that continuously as this pollen tube is elongating. Okay. In the next stage, let us say now if this is the same thing. If the pollen tube is now elongated, here and there at some points there will be one plug that is formed. We call this plug as a callus plug. Okay, we call it as callus plug. What is this callus plug? What is the function of this callus plug? This callus plug that is formed will not allow the vegetative cell or the contents of the vegetative cell and generative cell to be pushed back. All the content of this cell, the pollen grain, will be there present at the tip of the pollen tube. They will not be scattered throughout the cell or throughout the pollen tube. They will be at the tip of the pollen tube. How it is maintained? It will be forming callus plug. As this pollen grain, some more, as it is increasing in length, okay, as it is increasing in length, here again there will be the callus plug that will be formed. Keeping in the generative cell and vegetative cell at the tip of the pollen tube, and you would recall that we already discussed that about 60% of the angiosperms, if you are seeing 60% of angiosperms, okay, if you are seeing, they will be releasing the pollen grains. Okay, 60% of the angiosperms will be releasing the pollen grains at two-celled stage, which we are seeing now at two-celled stage. And the other percent of at least 40% of those angiosperms, 40% of the other angiosperms will be releasing the pollen grains at three-celled stage. Why these 2 points are very important points? Because once the pollen tube is formed, okay, as it is moving towards the ovule, we should remember that if it is two-celled stage, it will change into three cells. Means here, if you are looking at the generative cell, okay, that generative cell that is there here, say suppose if this is the pollen tube, here is what vegetative cell we are saying, here is what generative cell, this is what now two-cell stage. That is two-cell stage. But as the pollen tube is formed, reaching the ovule, during that period of time, during that process, that generative cell will undergo mitotic division. The generative cell will undergo mitotic division. And divides into two nuclei. It divides into two nuclei, we say. And what is the other one? It is the generative cell. And here are two now. Actually, we said how many are there? One generative cell and one vegetative cell. But here what we are again saying, there are two cells here now. When this will be seen, when the pollen tube is germinating, then the generative cell divides into two by mitosis. But in some other plants, suppose now the other 40% of the angiosperms will be showing three-cell stage. These are all three cells now. which are already present in the pollen grain, which are already present in the pollen grain, we say. Okay, there are 3 cells now. Okay, vegetative cell and 2 of the male gametes. Already there will be 3 cells that will be formed before the pollen tube is germinating on the stigma or into the style. But most of the cases we have to remember that the 60% of the angiosperms will be released in the pollen grains at 2 cell stage, and when the pollen tube is formed, inside the pollen tube, that generative cell will divide by mitosis and forms 2 cells. Whereas in the other case of the other 40% of the angiosperms, already the division is occurred and there are already what 3 cells that are present. There are 3 cells that are present. Now this pollen tube reaches to what now? The ovule. Reaches the ovule to enter into the ovule. To enter into the ovule. So now this pollen tube is having how many cells now? It is having 3 cells, we say. One is the vegetative cell. The other 2 are the 2 male gametes, we say. 2 male gametes. And as it moves here, it is having cytoplasm of the vegetative cell. And as it moves here at the tip, where is now the pollen tube that is present? It is present at the micropyle region of the ovule. This is the common region that we talk about. The pollen tube will be entering into the ovule through what? Micropyle. It will be entering into the ovule through what? Micropyle. But otherwise, there are 3 ways. There are 3 ways through which the pollen tube can enter into the ovule. There are 3 ways through which the pollen tube can enter into ovule. So we are saying that the pollen tube that grows and develops through the stigma, style, reaches the ovary and enters into the ovule. This is the ovary and this is the ovule at the center. So as it enters into the ovule, mostly this pollen tube enters through the micropyle region. But otherwise, there are various ways the pollen tube may enter into the ovule. So we can see here that when the pollen tube, if this is the pollen tube, okay, when the pollen tube enters through the micropyle region, when the pollen tube enters into the ovule through the micropyle region, we call it as porogamy. We call it as porogamy. So what is porogamy? When the pollen tube is entering into the ovule through the micropyle region, we are calling it as the porogamy. This is a common one. But otherwise, there are also some other ways the pollen tube may enter inside into the ovule. See here, we know that when we draw the ovule, this region of the ovule, we call it as what? Chalaza. Okay, the basal part of the ovule. The basal part of the ovule is chalaza. So sometimes when the pollen tube grows, it may enter through the chalazal region also. It may enter through the chalazal region into the ovule. So when the pollen tube is entering through the chalazal region, okay, through the chalazal region, and then comes and reaches this region where there is egg, we call this as what? Chalazogamy. Chalazogamy. Here the word itself tells us to keep it in our mind. So if the pollen tube is entering through the chalazal region, it is called as what? Chalazogamy. We are calling it as chalazogamy. So here the pollen tube enters through the chalazal region. And the third one is called as mesogamy. The third one, let us say C, it is called as mesogamy. So what happens? How the pollen tube enters into the ovule in the case of mesogamy? In the case of mesogamy, the pollen tube may enter. What are these? These are the integuments. These are the integuments. So the pollen tube may enter into the ovule through the integuments, crossing outer integument and inner integument. So when it is entering through the integuments, we call it as what? Mesogamy. Otherwise, it is also the pollen tube may enter through the funicle region, funiculus. When the pollen tube is entering through the funiculus also, we call it as what? Mesogamy. So there are 3 ways through which the pollen tube may enter into the ovule. One is called as what? Porogamy. So in case of porogamy, what happens? The pollen tube enters through the micropyle region. Second one is what? Chalazogamy. when the pollen tube is entering through the chalazal region, the basal part of the ovule. The third one is what? Mesogamy, when the pollen tube is entering through the integuments of the ovule or through the funiculus of the ovule, we call it as what? Mesogamy. So in all these three ways, the pollen tube can enter into the ovule. So we studied that the pollen tube will be entering into the ovule in three ways. One is what? Porogamy. Then the other one is what? Chalazogamy. And the other one is mesogamy. So here the best examples that are undergoing through the pollen tube entering into the ovule through the micropyle region is porogamy, we said. The best example is what? Lily. So in lily, the pollen tube will be entering through the micropyle region. And in the chalazogamy means what we said? When the pollen tube is entering through the chalazal region, the basal part of the ovule. When the pollen tube is entering through the chalazal region, we are calling it as chalazogamy. Best example is what? Casuarina. And mesogamy. Mesogamy means what? When the pollen tube is entering either through the integuments or the funiculus of the ovule. That is called as what? Mesogamy. Best examples are cucurbita and populus. These are the two examples which we should keep in mind when we discuss about the porogamy, chalazogamy and mesogamy. So we said that the pollen grain that is falling on the stigma germinates and the pollen tube extends, elongates and reaches to the ovule, to enter into the ovule where there is egg inside the embryo sac, So this pollen tube is bringing what cells? The vegetative nucleus, the generative nucleus. Two male gametes are brought. As soon as the pollen tube reaches the micropyle region, the vegetative nucleus disappears. Leaving behind two male gametes. There will be two male gametes that will be there. Now these two male gametes will be entering into the ovule, through the micropyle region. So at the micropyle region, the embryo sac will be consisting of a structure called as egg apparatus. What is this egg apparatus? The egg apparatus will be consisting of two cells called as synergids. They are called as synergids. Two cells called as synergids and one cell which is very, very important, which we call it as the egg. So these three cells, two synergids and one egg, is called as what? Egg apparatus, which are there in the embryo sac towards the micropyle of the ovule. This egg apparatus is present where? It is at the micropyle region of the ovule in the embryo sac. Now in this egg apparatus, we are coming across synergids. So if you look at the synergids, they are like pointed beaks. So these synergids will be having extensive infoldings. They are having extensive infoldings. These extensive infoldings present in the synergids is called as what? Filiform apparatus. So what is filiform apparatus? They are the extensive infoldings. Present in the synergids. They are at the base. And this filiform apparatus, it is extensive infoldings. Okay, extensive infoldings, which are called as filiform apparatus present in the synergids. The word synergids means working together. The word synergids means working together or works together. This is the meaning of the word synergids, works together. So they are having what? Filiform apparatus. It is a highly folded wall that is there. It is called as filiform apparatus. Please remember, this filiform apparatus is under the formation of the filiform apparatus is under the control of genes, where the genes will be producing one protein called as MYB98 protein. This will be helping in formation of the filiform apparatus. filiform apparatus. So what is the function of this filiform apparatus? It helps in attracting the pollen tube. It helps in attracting the pollen tube towards the egg. So here in this diagrams we can see very clearly. Now this is highly viewed diagram showing how the pollen tube. This is what the pollen tube. This is the pollen tube. The pollen tube is entering through the micropyle region. The pollen tube is entering through the micropyle region. How it is entering means it is crossing what? Filiform apparatus of what? Synergids. There will be egg here. This is the egg. And these three together, what we said? We call it as egg apparatus. So now see here. When the pollen tube enters, the male gametes will be attracted to enter into one of the synergids. This is very important point. They are not directly fusing with the egg. To enter inside into the embryo sac where there is egg, they are attracted by the filiform apparatus. It is believed that the filiform apparatus will be producing some chemicals to attract this pollen tube. So the pollen tube is attracted towards the synergids. Which are consisting of filiform apparatus. So remember that filiform apparatus attracts the pollen tube. So the pollen tube now reaches the synergids. These two are the synergids. So once this pollen tube reaches the synergids attracted by the filiform apparatus, now the pollen tube ruptures. Releases the male gametes inside. Here, now the male gametes carried by the pollen tube are released into one of the synergids. Once these male gametes are released into the synergids will guide these male gametes to fuse with what? One male gamete to fuse with the egg and another male gamete to fuse with the polar nuclei that are there in the central cell of the embryo, of the ovule. The central cell will be having the polar nuclei. So one male gamete will go there and fuses, whereas one male gamete will fuse with the egg. One male gamete will fuse with the egg and another one will be fusing with the central polar nuclei present in the central cell of the ovule. So this is how the male gametes are brought by the pollen tube to enter inside into the embryo sac and fuse with the egg. So here, from the time the pollen grains falls on the stigma, means what type of pollen grains? Compatible pollen grains, means right type of pollen grains. From the time pollen grains falls on the stigma until they reach the ovule, all these events that are taking place are coming under as what? Pollen pistil interaction. So the events that are taking place from the time the pollen grain falls on the stigma until it reaches the ovule is nothing but called as what? Pollen pistil interaction. Already we said that because of the pollen pistil interaction, what happens in the pollen pistil interaction? 2 points that we have to remember is that in the pollen pistil interaction, it will recognize the right type of pollen grain. Very important. In the pollen pistil interaction, it will recognize the right type of pollen grain and it will reject out the wrong type. It will reject if it is wrong type. Means what? Incompatible ones will be rejected. Only it will promote the growth of the compatible pollen grains on the stigma to grow, elongate with the help of the pollen tube and reach the ovule to enter into the embryo sac where there is egg. So from the time it falls, what is that? The pollen grain on the stigma until it reaches the ovule is called as pollen pistil interaction. So this knowledge of pollen pistil interaction is very useful for the plant breeders, even in altering or in manipulating the incompatible pollination, even in altering or manipulating the incompatible pollination. This knowledge will be very useful for the plant breeders so that they can get desired hybrids, or superior varieties varieties. So, in conclusion, we will try to understand the process of pollen germination. In conclusion, we will try to understand the process of pollen germination. How the pollen grains can be germinated. You can do this by yourselves in the laboratory. So, what are the steps that we have to follow? We will just see. So, here in the process of pollen germination, we have to collect the pollen grains. Pollen grains of different plants. For example, here a pea plant or a chickpea or Protellaria or balsam or vinca. Very freely available for us is the vinca or the pea plant and the chick plant. Of course, most of them are maybe based on different regions. You will be getting these plants. So, based on that, when you collect that flower of the plant, you have to dust the pollen grains on a slide. Okay, you have to take a glass slide. We are talking about the process of pollen germination. So, you are collecting the pollen grains, for which you need a glass slide. On the glass slide, you will dust the pollen grains. You will dust the pollen grains. After dusting the pollen grains on the glass slide, add a drop of sugar solution of about 10%. 10% sugar solution should be added on the pollen grains. So when you add this, we already studied here on the stigma what we said. This stigma will be having water, it will be having sugars and also organic acids. And this water, sugar or organic acids will be absorbed by the pollen grains and it will expand and produces a pollen tube. So in the same way, here what we have to remember when we are talking about this is, please remember, growth of the pollen tube is chemotropic. Growth of the pollen tube is chemotropic. Please remember this. Growth of the pollen tube is chemotropic. Okay, chemotropic. So here means what? It is under the control of what? Chemicals. It is under the control of chemicals. So keeping that in mind, we are using that in our practical now. Okay, practically we are trying to understand through a process now how the pollen germination will be taking place. So what we are taking, a glass slide, dust the pollen grains of any of these plant flowers. Okay, so once you dust them, add a drop of 10% of sugar solution and leave it for how many minutes? About 15 to 30 minutes you leave it without disturbing it. After leaving it for 15 to 30 minutes, when you see it under the lower lens of the microscope, okay, when you see it under the lower lens of the microscope, you will be seeing the images like this, where the pollen grain is producing what now? It is producing the pollen tube. So this is how we can do a practical. Okay, practically we can do this in the laboratories to prove how the pollen grains will produce the pollen tube. Okay, that we are calling as what? The process of pollen germination. Process of pollen germination. Thank you so much.